Next we have PPREV 133, Abdeali. Abdeali, I've already made you a panelist. Please open your webcam and start your presentation.
Am I audible, ma'am?
And yes. And one more announcement. I would request everyone to stay till the end, as a screenshot will be taken at the end. So please stay till the end. Yes, Abdeali.
Good evening, respected panel members. This is Abdeali Hargarwala from Mallareddy Pharmacy College, Secunderabad. Today, I am here to present my poster entitled The Versatility of Marine Drugs in front of you. The reason for selecting this particular topic for my presentation is mainly due to the tremendous potential and unexplored diversity of marine organisms that can significantly aid in revolutionizing the new era of medical sciences. Over the last several decades, the vast majority of the human population have been facing various health-related ailments. For some, there are certain specific treatments where the others can only be diagnosed and treated to certain extent, such as cancer, AIDS, osteoporosis, HIV, etc. As we know, marine biosphere is one of the largest of the Earth's biosphere and comprises enormous number of marine organisms. This rich diversity serves as a source of unique chemical compounds that can help in huge perspective for the development of pharmaceuticals, cosmetics, nutritional supplements, molecular probes, and also as a diverse resource for new drugs for combating many major diseases. The versatility of the oceans depends mainly upon the realm of survey and experimentation. Many marine products which are unknown to the mankind are the key for the development of new drugs and secondary metabolites. There are a number of sources for marine drugs such as sponges, tunicates, echinoderms, coelantrates, and molluscans, seaweeds, marine algae. Also, the marine drugs can be classified according to their pharmacological activity into antiviral, antibacterial, antifungal, antiprotozoan, anti-inflammatory, anti-hyperlipidemic, and antioxidant. Ziconotide is the first drug of marine origin isolated from a toxin of conical cone snail. It was approved by US FDA on 31st December 2004, and it is reported to be the most powerful painkiller till date. It is administered intrathecally and it acts by blocking the N type of calcium channels which is present in the primary nerves of spinal cord. Coming to the isolation of marine pharmaceuticals, there are a number of steps being followed, starting with extraction, chromatographic purification, dereplication, structural elucidation, and bioassay testing. In the first step of extraction, the sample is either triturated or extracted with a suitable solvent system, such as methanol chloroform, ethanol acetonitrile, etc. Then the sample is dried using lyophilization technique, which is also known as cryodesiccation or freeze drying, which is nothing but a low temperature dehydration process. Then the chromatographic purification is being done using various chromatographic techniques. There are also novel approaches being made for the extraction of these novel compounds, such as solid liquid extraction, ultrasound assisted extraction, subcritical water extraction, and pressurized solvent extraction procedure. For the structure elucidation, we perform high resolution mass spectrometry, and for sometimes tandem mass spectrometry is also performed for large molecules such as glycolipids, polysaccharides, proteins, etc. Now, coming to the role of marine drugs in COVID-19, there had been a recent article published by a group of researchers at Reliance Research and Drug Development Center in Navi Mumbai that marine red algae, which is called Pyridium species, containing the sulfated polysaccharide, can prevent the spread of coronavirus. Sulfated polysaccharides such as carrageenans are the selective inhibitors of both enveloped and non-enveloped viruses, and they act by preventing the binding or internalization of virus onto the host cell. As we know, there will be again certain challenges which will be resulting in the limited supply of the marine pharmaceuticals. As the marine organisms are closely associated with the symbiotic association, it is very difficult to extract them. Also, there are toxicity study issues. And coming to the global market, America is held the largest of over 41% share among all the global countries which are producing the marine pharmaceuticals. There are also various institutes in India engaged in developing the marine pharmaceuticals, especially the Bose Institute at Kolkata. Now, I would like to conclude my presentation by saying that marine pharmaceuticals bring about a new hope for the future drug development, and the evolution of marine pharmacology as a specialty in India will also help us in optimizing the use of the vast coastline of our country for the future prospects. And there is always a history hidden, there's a mystery hidden in the chemistry of the oceans. Thank you so much.
Yeah. Thank you, Nagar Garwala.
Thank you, sir.
Yeah, your presentation is good, and your designation.
Sir, I am from B Pharmacy Department, fourth year, sir, final year.
Final year student. Okay, good. So, give me some examples of marine drugs.
The marine drugs such as citarabine, which is isolated from sponge, it can be used in the treatment of cancer, and also omega-3 fatty acids, which are usually isolated from the fish, can be used in the treatment of hypertriglyceridemia, and macrolide lactones, which are isolated from the bryozoans, can also be used in the treatment of Alzheimer's disease.
Okay. So, give some examples that of solvents that are used in the extraction of marine drugs.
So the most commonly solvent used is carbon dioxide because it is also cheap and it is widely available, sir. And other solvent system used for the extraction are methanol chloroform and acetonitrile ethanol.
Okay. Give some methods of extraction of marine drugs.
So the methods of extraction widely used are solid liquid extraction, microwave assisted extraction, ultrasound assisted extraction, pressurized solvent extraction. These are certain novel approaches which are being made for the extraction of marine pharmaceuticals.